Bismillahirrahmanirrahim. Assalamu alaikum. This is me, Shumaila Qadir, conducting your course 317. Today we have our topic, gluconeogenesis. What is gluconeogenesis? It is the formation of glucose or glycogen from non-carbohydrate sources. So somewhat it is the reverse of glycolysis, which was the breakdown of glucose. But here, in the process of gluconeogenesis, the formation of glucose or glycogen is occurred from non-carbohydrate sources. The process of gluconeogenesis is so important. So why it is necessary in the body? First, the gluconeogenesis is required to meet the requirements of glucose in the body. When the carbohydrates are not available in sufficient amount from diet, so non-carbohydrate sources are utilized and glucose or glycogen is formed. And this is occurred to provide energy for nervous system, for RBCs, etc. And also it maintains the intermediate of TCA cycles. It also provides as a source of milk sugar, that is lactose, for lactating mammary gland. And the second, the gluconeogenic mechanism are also required to clear the products of metabolism of other tissues from the blood. For example, the lactic acid which is produced from the muscles and RBCs, they are involved in the process of gluconeogenesis. And secondly, the glycerol that is produced by adipose tissues by the process of lipolysis of triacylglycerol. Side of gluconeogenesis, where the process of gluconeogenesis is occurred. In mammals, the principal organs which are involved in the gluconeogenesis are liver and kidneys, because they possess the enzymes required for the process of gluconeogenesis. Now come to see the substrates which are required for gluconeogenesis. So the principal substrate are glucogenic amino acids, lactates or pyruvates, and glycerol. The amino acids which are involved in the process of gluconeogenesis are pyruvate forming amino acids, and those which are produced as intermediates of TCA cycle. For example, oxaloacetic acid and alpha ketoglutaric acid. Whereas lactate and pyruvate are also involved in the process of gluconeogenesis, which are interconvertible to each other. Third, glycerol. The glycerol also participate in the process of gluconeogenesis, which is obtained by the process of or from the process of lipolysis of fats. In the process of gluconeogenesis, the pathways involved include glycolysis. This is here the reversal of glycolysis, and citric acid cycle, and also some other special reactions. The glycolysis was the, as I told you, it is the lysis of glucose. So the reversal of this glycolytic pathway is contributing to gluconeogenesis, formation of glucose or glycogen. So the main pathway of gluconeogenesis is a reversal of glycolytic pathway. But here certain barriers are there, because recall the glycolysis, there are 4 types of reactions which are irreversible. So the thermodynamic barriers prevent the simple reversal of glycolysis at certain points. So special adaptations are required to form glucose or glycogen. What are these barriers? Number 1 reaction is between pyruvic acid and phosphoenol pyruvate. The second reaction is between fructose 1,6 biphosphate and fructose 6 phosphate. So reversal from fructose 1,6 biphosphate to fructose 6 phosphate is not possible and requires certain adaptation. Third, the reaction between glucose 6 phosphate and glucose require special adaptation or modification to continue the formation of glucose. The last one is the reaction between glucose 1 phosphate and glycogen, because glycogen is formed from glucose, so also required certain adaptation or modification. Now see here, it is simply the glycolytic pathway. It is not the gluconeogenesis pathway. But here I would like to make you understand where are these adaptations required. First, the reaction between pyruvic acid and phosphoenol pyruvate. You can see here, this reaction is not reversible in the process of glycolysis. Second, the conversion of fructose 1,6 biphosphate to fructose 6 phosphate is not possible. This reaction is also because the irreversible reaction of conversion of fructose 6 phosphate to fructose 1,6 biphosphate. Third, reaction of glycolysis, the formation of glucose 6 phosphate from glucose. This is a irreversible reaction. So conversion from glucose 6 phosphate to glucose is not possible. And the last is the reaction between glycogen to glucose 1 phosphate. This is also a irreversible reaction, and conversion of glucose 1 phosphate to glycogen requires certain adaptation. Now we are going to discuss first barrier, that is the reaction between pyruvate and phosphoenol pyruvate. So what are the adaptation required? We will see here. The conversion of pyruvic acid, which is interconvertible to lactic acid, require 2 enzymes to convert to phosphoenol pyruvate. First, pyruvate carboxylase, which is a mitochondrial enzyme. The second is phosphoenol pyruvate carboxykinase. This is cytosolic enzyme. So the first reaction is the conversion of pyruvic acid to oxaloacetic acid, and this requires the enzyme pyruvate carboxylase, which is mitochondrial enzyme. So in presence of ATP, biotin and carbon dioxide, pyruvate is converted to oxaloacetic acid, and here ATP is converted to ADP with 1 phosphate group. The second reaction convert the oxaloacetic acid to phosphoenol pyruvate and require the enzyme phosphoenol pyruvate carboxygenase. This is cytosolic enzyme and require GTP or ITP, which are converted to either GDP or IDP. GDP from GTP and IDP from ITP with vibration of carbon dioxide. After the formation of phosphoenol pyruvate, the other reaction will be continued in the reversal manner of glycolytic pathway, because from there start reversal reaction of glycolytic pathway. Now the important thing, the form oxaloacetic acid, which is formed inside the mitochondria, and the other reaction, the conversion of oxaloacetic acid to phosphoenol pyruvate, is in cytosol. Oxaloacetic acid has to cross mitochondria, and this oxaloacetic acid is impermeable to mitochondrial membrane. So what happened then? By two mechanisms, its transfer is occurred to cytosol. The first, the oxaloacetic acid in presence of enzyme malate dehydrogenase converted to malic acid. And malic acid has ability to cross mitochondrial membrane, and then this malic acid again convert to oxaloacetic acid in presence of enzyme malate dehydrogenase. Now this oxaloacetic acid is converted to pyric acid and simple reversal of glycolysis process is continued. And the second mechanism is the formation of citric acid. The oxaloacetic acid combined with first acetyl-CoA to form citric acid that you saw in TC cycle, and this citric acid is permeable to mitochondrial membrane, and citric acid is then cleaved by citric acid cleavage enzyme and reform again oxaloacetic acid to form pyric acid and to continue the process of gluconeogenesis by the reversal of glycolysis. Another important reaction is that pyric acid can also be converted to oxaloacetic acid by the formation of malic acid. So the conversion of pyric acid to phosphoenol pyruvate can be summarized in the figure here. This is the pyric acid, and it has to be converted to phosphoenol pyruvate. How this occurs? Because here the reaction, the glycolytic reaction, phosphoenol pyruvate is converted to pyric acid. This is irreversible reaction. So here the pyric acid cannot convert back simple by this reaction to phosphoenol pyruvate. So what happened? It can either form oxaloacetic acid by the enzyme pyruvate carboxylase, which we discussed, and it can also first convert it to malic acid by the enzyme malic enzyme. This reaction is reversible reaction, and then this malic acid is converted to oxaloacetic acid by the enzyme malate dehydrogenase, which then by either route, the oxaloacetic acid which is formed is then converted to phosphoenol pyruvate and will continue the process of reversal of glycolysis or the process of gluconeogenesis, the formation of glucose. Now the second barrier or the reaction of glycolysis, where reconversion of glucose 6-phosphate from fructose 1,6 biphosphate is not possible, or the reaction is irreversible in glycolysis. So what adaptation is required between the fructose 1,6 biphosphate to form fructose 6-phosphate? This fructose 1,6 biphosphate will form fructose 6-phosphate in presence of enzyme fructose 1,6 biphosphatase. And this is the key enzyme of gluconeogenic pathway, and thus it determines whether or not a tissue is capable of forming glucose or and glycogen, not only from pyruvic acid or lactic acid, but also from triose phosphates. Where the enzyme is present? It is present in liver, kidneys, and intestine. And it is also present in striated muscles, but it is absent in adipose tissues, cardiac muscles, and smooth muscles. The third barrier is between glucose 6-phosphate and glucose. So the formation of glucose from glucose 6-phosphate requires enzyme, that is glucose 6-phosphatase, which convert glucose 6-phosphate to glucose. And this enzyme is also present in liver, kidneys, intestine, and platelets, whereas it is absent in adipose tissues and muscles. And as it is absent in muscles, so it cannot be able to contribute to blood glucose directly by the process of glycogenolysis. The fourth and last barrier is in the formation of glycogen. As I told you, that gluconeogenesis is the process of formation of glucose or glycogen from non-carbohydrate sources. So the conversion required certain adaptation to form glycogen. In the previous lecture, we also have covered the process of formation of glycogen, that is glycogenesis, and it required the enzyme glycogen synthase and so on. The breakdown of glycogen to glucose 1-phosphate required enzyme phosphorylase, but reconversion is not possible here and required certain specific adaptation, which you have already seen in the process of glycogenesis, which involve the different pathway through the formation of UDP, glucose formation, and the activity of glycogen synthase. Now see here the four barrier or the adaptation followed in the process of gluconeogenesis for the formation of glucose. So first, you can see here the conversion of pyruvate to phosphoenol pyruvate is occurred by the formation of oxaloacetic acid. The pyruvate form oxaloacetic acid, which can either by formation of malic acid or citric acid, then release from mitochondria to cytosol, and this malic acid again form oxaloacetic acid, and citric acid also will be able to form oxaloacetic acid, and ultimately it will form phosphoenol pyruvate. And not only by the enzyme pyruvate carboxylase, the pyruvate can also form malic acid first and then form oxaloacetic acid, And also pyruvic acid will form malic acid by malic enzyme, and then it will form oxaloacetic acid by malic dehydrogenase enzyme, and then finally the oxaloacetic acid will form phosphoenol pyruvate, which will then convert it to 2-phosphoglyceric acid, which will continue the reversal of glycolysis pathway. And the second barrier which occurred in the reversal of glycolysis pathway, it's the conversion of fructose 1,6 biphosphate to fructose 6-phosphate. And this is occurred by the enzyme fructose 1,6 diphosphatase. So in this way, the formation of fructose 6-phosphate is possible here. The other side in the figure showing the reaction of glycolysis, where fructose 6-phosphate form fructose 1,6 diphosphate in presence of enzyme phosphofructokinase and ATP. So this was the second barrier. Now the third barrier is in the formation of glucose from glucose 6-phosphate. Because in glycolysis pathway, glucose is converted to glucose 6-phosphate by the presence of enzyme glucokinase specific to liver or hexokinase. This is present in other tissues. But here in the process of gluconeogenesis, the enzyme required for the conversion of glucose 6-phosphate to glucose is glucose 6-phosphatase and water molecule. So here also the last reaction is remaining, which is the formation of glycogen from glucose 1-phosphate, and in glycolysis, or it required the conversion of glycogen to glucose 1-phosphate by phosphorylation or by the enzyme phosphorylase, but the reversal required process of glycogenesis that you already studied in the lecture of glycogenesis. So these were the all 4 barriers in the glycolysis pathway, which require certain adaptation to form glucose or glycogen. The amino acid form either pyruvic acid or intermediate of TCA cycles after transamination or deamination, which will continue the process of gluconeogenesis. After the gluconeogenic amino acid, the lactic acid also participates in the formation of gluconeogenesis because it is converted to pyruvic acid, which enters the mitochondria before conversion to oxaloacetic acid, and ultimately by the process of reversal of glycolysis, glucose formation is occurred. So the net conversion of both gluconeogenic amino acids and lactic acid via formation of pyruvic acid to glucose or glycogen constitutes the principal and major glucogenic pathway. The reversal of glycolysis pathway is the major pathway of gluconeogenesis. Here you can see the lactic acid can form pyruvic acid, which can further leads to the formation of oxaloacetic acid, and then phosphoenol pyruvate is formed and reversal of glycolysis pathway is continued and glucose formation is occurred. And also by gluconeogenic amino acid, the formation of oxaloacetic acid is occurred, which then converted to phosphoenol pyruvate, and the process of reversal of glycolysis occurred for the formation of glucose or glycogen.